So now we are coming to the fourth topic, which is secondary growth.
Okay.
So we have already studied that secondary growth generally take place in dicot plants, dicotyledon plants.
In monocotyledon plants, only primary growth will take place.
Yes.
Okay.
In dicot plants, secondary growth will take place, and this secondary growth is mainly in dicot plants, and this is generally known as the increase in girth. Girth is nothing but the lateral sides of the stem.
Surrounding sides.
Yes.
Okay.
If you see banyan tree, it will be very big, right?
Yeah.
So it is increasing in girth, like this. It is increasing. Yeah. So that is
its
increasing.
Surface area is larger. It's increasing its size.
Yes, it is from this stem. It is increasing like this.
Yeah, that's only.
And here the tissue,
okay,
which is involved to increase its lateral growth are two lateral meristems.
Okay.
So the first one is vascular cambium, and the second one is cork cambium.
Okay.
So here first we are going to discuss about vascular cambium.
Okay.
So in young stems, this secondary growth. In young stems, this secondary growth will be This meristems, these lateral meristems will be like patches. They will be very small.
Very small patches it will be.
So when they mature, these will become enlarged and they will grow into vascular cambium and cork cambium.
Patches to vascular cambium or cork cambium or
vascular cambium. Both lateral meristems are present few in amount.
Okay.
So this vascular cambium, which are present few, it will become vascular cambium, and which are cork cambium, which are present few, these will become cork cambium.
Okay.
So now we are going to discuss about vascular cambium. So
cork
here
cambium in the next slide.
Yes.
Okay.
No, not in the next slides. We will be discussing some categories in this, and then we will be discussing about cork cambium.
Okay.
So here now we are going to discuss about vascular cambium. So it is a meristematic layer. So this layer, what it does, it cuts xylem and phloem. So xylem will be here, phloem will be here. So it cuts in between. Wherever the cuts are there, it is known as vascular cambium, and it is developed, and we are going to study the structure of them. And then in young stems where primary growth occurs, these are present in patches, that is in single layer it will be present. And generally primary growth takes place by the shoot apical meristem and root apical meristem. They are responsible for primary growth. But here for secondary growth, the lateral meristems are responsible for secondary growth. Okay. So under vascular cambium, we are going to study the formation and activity of cambial ring, how it is forming, and what is the activity of that ring, that what we are going to study. And then we are going to study about spring wood and autumn wood, and heartwood and sap wood. Okay, so these four topics we are going to discuss in detail about.
So the meaning of these words we will be discussing in the next slide.
Yes. Okay. So in vascular cambium, first we are going to discuss about formation of cambial ring.
Formation of cambial ring. Okay.
So in dicot stems we have already seen that cambium is present between xylem and phloem. We have already discussed. Yes.
It forms like a patches and grows very fast.
First it forms in patches, and then it forms for secondary growth. Correct only. So it is present in between xylem and phloem. Okay. So here this is known as intrafascicular cambium, which is present here. This gap is there, right, between xylem and phloem. It is intrafascicular cambium.
Okay.
And here this cambium connects the other cambium.
Like this it comes.
Yes.
Okay.
So this is known as in between the two xylem and phloem. So in between this circle is known as interfascicular cambium.
In that it will not connect. In this it will connect.
No.
So both are same only. Interfascicular cambium, intrafascicular cambium. So what is the difference between them?
It is inside between xylem and phloem. It is outside between xylem and phloem.
Oh, it's present here. It's not present here.
Yes.
Okay.
So it joins, it connects. Okay. And here, what is the activity of cambial ring? How does it divide? Okay. So this cambium, this cambium is a ring-like structure. Totally this cambium. will divide inside as well as outside.
Okay.
On its inner side and as well as its outer side. So this is known as xylem.
Yeah.
So when this cambium divides inner side towards the xylem, it forms secondary xylem.
Secondary xylem.
Yes.
When it divides into
inner side of the xylem. So this, if you take this is xylem, so when this cambium divides into inner side of the xylem, so this is known as secondary xylem.
Okay.
And when this cambium divides outer,
It's called secondary phloem.
yes, towards the phloem, it is known as secondary phloem.
Towards the phloem, it's called secondary phloem.
Yes.
Towards the xylem, it's called secondary xylem. That's it. Okay.
So now inside what is there?
Inside
Xylem, here xylem.
Yeah.
Secondary
secondary
xylem.
xylem, secondary phloem,
And then phloem.
phloem,
So first what? Xylem.
xylem,
And then
secondary xylem,
and then
phloem,
secondary phloem and then only phloem, because it divides here and here. Okay. So this is the structure generally, and these cambium cells, they divide more towards the xylem
more towards the xylem
than towards the phloem. So therefore secondary xylem is present more in amount in stems.
So you are dropped on more amount. Okay.
So they are present more in amount. And this phloem and secondary phloem, no, they come when these cells divide and divide. These will form the upcoming tissues under epidermis. So they will be the outer layer, and they can get fused sometimes. Both phloem and secondary phloem, sometimes they get fused.
Okay.
So this is about the activity, how it divides the cambium ring. Okay. So next we are going to see about springwood and autumnwood.
Okay.
So after activity of cambial ring, so now we are going to see about spring and autumnwood.
Okay.
So what is generally spring and autumnwood? Springwood and
season. Autumn means autumn season.
Yes.
So what do you mean that wood?
Springwood is nothing but the tissue which is forming in spring season.
Spring season. Autumnwood is, that is creating on autumn season. Okay.
Yes. So here it is based on the activity of cambial ring.
Activity of cambial
ring.
Okay.
So cambial ring we have studied now, which divides to the inner side of the xylem, forms secondary cambium. Outer side of the towards the phloem, it forms a secondary phloem. So here this activity of cambial ring is based on various environmental factors.
Okay.
Okay, like temperature, light intensities and everything. So here now we are going to see the temperature and the environment factors, like spring and autumn. In spring what will happen, and in autumn what will happen. So in spring season, this cambial ring, it will actively divide. So it is known as active cambium. It will actively divide cells towards the xylem. Mainly it produces large number of xylary elements. Large number of xylary elements. So this is known as this wood generated during spring season.
Xylem elements are produced large.
Yes.
Okay.
So at this season, this tissue is known as the wood developed in this season is known as spring wood or early wood.
Okay.
So this is about spring. In spring what will happen, and in winter it produces less xylary elements. Okay. So that is, it will produce less secondary xylem.
Okay.
So since therefore it is known as a less active cambium. It is less active in winter season. So this is known as autumn wood or late wood.
Okay.
Okay. So this is generally the two woods. So this spring wood, it will be light in color and it will have low density.
Okay.
And autumn wood will be dark in color and it will have high density. So the combination of these both rings forms annual rings.
Okay.
So if you cut a stem like this, there will be many rings, right?
Yeah.
Light, dark, light, dark. If you count all the dark rings, you can estimate the age of tree. How years the tree has been, what is the age
From
of
what,
tree?
which age they have
Grown. Yes. So you can estimate the age of tree by using these two rings, which are known as annual
So we
rings.
should combine both this.
Yes.
And we should estimate the value in it as.
No, you have to not combine.
Okay.
One will have a light, one spring wood is light in color, and autumn wood is dark in color.
So, you should
autumn wood
6
will
months,
be
6 months you should take it.
no. Autumn wood will be dark in color, so therefore it appears like lines. So you will count all the lines, which is autumn wood.
If you count all the lines in what, if one line is equal to
6 months.
6 months. That's what I'm saying.
One light see here. So consider this as this white color as spring wood, and this blue color as autumn wood.
Okay.
So these are generally rings. So how will you count?
So I'll count this one, this 6 months, 6 months, 6 months.
How? No.
Then?
This white is forming 6 months.
Okay.
This is forming 6 months. So totally 1 year.
Totally 1 year.
And this white is formed 6 months. This is formed 6 months, 1 year.
So totally 2 years. Then 3 years.
Yes.
Okay.
So which you will count only these lines. Dark lines you will be counting. Okay. So if you count 6 months, it will not come correct. This dark lines you will be counting 1 year, 1 year. It is just an estimate age of tree.
It may be 1 year, 1 year more or 1 year less.
So next we will see about heartwood and sapwood. So now we are going to discuss about heartwood and sapwood.
Okay.
So in old trees, the secondary xylem, which is formed by cambium, it is dark brown in color. Okay. So this secondary xylem is made up of deposition of tannins, resins, oils, gums, and etc. So it is produced by the secondary xylem. Okay, in older plants. So here these substances make the plant generally hard, and these substances even make the plant to become resistant to insects and other microorganisms and etc. And here this region, which is composed of tannins, resins, oils, and gums, which is the secondary xylem, this region is composed of dead elements and highly lignified cells. So lignified is nothing but the cells composed of lignin. Lignin is a material. Okay, it is composed of lignin. We have already seen this lignified, and this type of wood is called as heartwood, which contains these everything and dead elements and deposition of tannins, resins, oils, gums, and dark brown. These are dark brown in color. So these are known as heartwood. And this heartwood, it does not conduct water from root to the stem. It just gives the mechanical strength to plants. Okay. And then the peripheral region to heartwood. If this is the heartwood, the outer region of the heartwood is known as sap wood, and it conducts water from roots to leaves, and it is light in color. Okay, so this is generally about heartwood and sapwood, which is about secondary xylem. So secondary xylem, which are old and contains deposition of these substances and are dark brown, and these are highly lignified and having highly lignified walls are known as heartwood, and the periphery is known as sapwood. This heartwood does not conduct water, where sapwood conducts water. So this is about heart and sapwood. So now we will go to the third subtopic under secondary growth, which is second subtopic. So these all comes under the subtopic vascular cambium. So now we are going to the second subtopic, which is cork cambium. Okay, so that we will discuss in the next slide. So now we are going to see a subtopic under secondary growth, which is the second subtopic after vascular cambium, which is cork cambium.
Cork cambium.
Yes. So this cork cambium is nothing but generally if you see in plants, since the cells which is intravascular cambium starts dividing and forms secondary xylem and secondary phloem, so these continuously forms, and therefore after some period of time, the outer cortical cells, they start breaking off and they start losing their capacity. So there is a need for the formation of new cells to replace those cells. So the replaced cells are known as core cambium or phellogen. Okay. So this cambium or phellogen, this cambium forms that replaces that broken cells which are outside, cortical cells. Okay. So when this cambium also divides outer side and inner side. Okay, this cambium is at center and some cells divides outer side, some cells divide inner side. Okay. So the outer side with the cells divided outer side are known as cork or phellum.
Cork or phellum.
Phellum. And the cells which are divided inner side are known as secondary cortex or phelloderm.
Secondary cortex or
phelloderm.
Okay.
So therefore the three layers it is formed. Outer, which is cork or phellum.
Inner, it is
outer cork or phellum, and the middle one which is cambium or phellogen. So and then secondary cortex or phelloderm. So phellum, phellogen, phelloderm. Okay. So these are the three layers which are outside.
Phellum, phellogen, phelloderm.
Yes. Okay. So these are present. And all the three layers together called as periderm.
Periderm.
Yes.
So phellum, phellogen, periderm. These three layers all together called as periderm. Okay.
Okay. And since these three layers, they continuously divide and form a large big layer. So therefore the layers which are outside to these cortical cells they die and they will be slawed off. They die, they get died and they will be slawed off.
Like dried leaves.
Yes.
Okay.
So these generally happen in this outermost cells. So
Like
the next
skin also sometimes happens.
yes. So the next we are going to say about bark.
Bark.
So this bark is nothing but all tissues exterior to vascular cambium. Vascular cambium is nothing but both interfascicular and intrafascicular, that cambial ring. We saw, right, which is making a secondary xylem and secondary phloem. Secondary xylem to the inside and secondary phloem to the outside. So the bark is the tissues exterior to vascular cambium, the tissues exterior to that cambium, which comprises secondary phloem and periderm. Okay, it comprises secondary phloem and periderm. Bark is known as secondary phloem and periderm. All the tissues are known as bark. Okay. So this bark is it is known as bark. So when this bark is formed at the early stage of the season, when the starting of the season, it is known as early or soft bark.
Early or soft bark. Okay.
Yes. So when it is developed late in the season,
it is hard bark.
Late or hard bark. Okay. So this is generally about bark. And phellogen. So this phellogen, which is the middle layer, phellum, phellogen, phelloderm in periderm. So the middle layer phellogen, it starts dividing and gives some breaks at the center. So this breaks will rupture the epidermis, will rupture all the cells outside and become lenticels. Lenticels are nothing but, when you see a tree generally, there will be many there will be many humps, will go deeper inside like that. Like this, it will be like a broken tree outside. The outer tissue.
The bark around.
Yes.
Yeah.
The outer.
If we take it off, it will come out.
Yes.
Some medicines also prepared by those things.
Yes. So those little gaps in the stem, that is known as lenticels.
So if this is some bark, see the bark.
Bark is nothing but bark is bark is different. This one is different.
So these gaps are lenticels, you are saying?
Yes.
That only I am also saying.
Bark is different. This one is different.
Yeah, the bark is different. The space between the bark is called as lenticels. This space.
Yes.
That's what I am saying. Okay.
space between the bark, but not all together bark. It is formed by phellogen. Bark is nothing but the comprisement of periderm and secondary phloem. Periderm is nothing but phellum, phellogen, and phelloderm. All these together, and secondary phloem. All these together comprises bark. So you cannot say bar in between bark. You can say in between. In between phellum. So the phellogen only makes this lenticels. So this makes like a small gaps, it will come
Crack
out.
like
Yes, cracks. Yes, exactly, cracks in the outer trees. You can see like many cracks. So these cracks permit the exchange of gases from outside to the inner tissues. They permit the exchange
of this cracks.
Yes. So this is generally about core cambium. Okay. So next we are going to see about secondary growth in roots.
So that's the second subtopic.
Yes.
After that, some topic.
No.
That's it. The chapter is over.
So next, with the next subtopic, the chapter gets over. Secondary growth in roots. Now we are going to the second subtopic under secondary growth.
Okay.
So which is secondary growth in roots. So here secondary growth generally occurs in dicot roots and dicot stem. Only dicot
and dicot stem. Okay.
Secondary growth we have already discussed that only it occurs only in dicot plants. So here in dicot root, vascular cambium is developed here also.
Okay.
And this vascular cambium is secondary in origin. So it develops after the root get matures and becomes secondary. After primary stage, when it starts secondary stage, this vascular cambium is developed. So this vascular cambium originates just below the phloem bundles, phloem bundles, and above the xylem, which is above the protoxylem. Okay. So in some part of pericycle, it's called pericycle.
Okay.
So this vascular cambium originates there and will start dividing cells inside and outside, which is secondary xylem and secondary phloem, and it forms a ring-like structure as we saw in dicot stem. The same stages occur in these roots. So that's all.
That's all.
Yes. So it is similar to dicot stem. So only the origin is different and
modifications.
yes, they only they only appear in secondary.
In OG.
Yes. So those cells which are in stem the vascular cambium are originated like patches. We have studied.
Yeah. Then it grows.
Yes, but here it is totally secondary. Okay. It will grow only in the secondary period of the plant. Okay. So this is about secondary growth in roots. It is similar to that of dicot stem. Okay. So with this the chapter ends with 4 topics, and the last topic is secondary growth. And thank you for watching this video. If you like this video, please subscribe and click the bell icon. And if you have any doubts regarding this chapter, please leave them in the comment section.
And please share the video. Thank you.